So our next speaker is Dr. Jason Marvin from Mass General Hospital. He's going to be telling us about the role of the innate immune response in regulating regeneration of the adult zebrafish enthesis.
Awesome. So thank you for the introduction, and it's really a pleasure to be invited to speak about my postdoc work today. So just starting with the basics, what are tendons? Tendons are essential connective tissues. They connect muscle to bone. As shown on the left, when the muscle contracts, it transmits loads to the tendon in the middle, and this enables locomotion of the skeleton and provides joint stability. And so in the adult human body, we have as many as 4,000 tendons that regulate a wide range of physical activities, whether those be more subconscious in nature, such as blinking, to those that are more strenuous, that require more thinking, such as exercise and rock climbing. So to facilitate the high and frequent mechanical demands of these tissues, tendons exhibit a very unique extracellular or matrix or ECM environment that's composed primarily of highly aligned type 1 collagen fibers, which I think you can appreciate much more in the leftmost image, which is a multi-photon micrograph of second harmonic generation signal, which labels collagen ultrastructure. And interspersed between these collagen fibers are the resident fibroblast-like tendon cells or tenocytes that populate the tissue. And so the tendon doesn't just wrap into the bone seamlessly, it actually has a very complex gradation. So at this tendon bone attachment site called the enthesis, you have that gradual change in both the ECM composition and the structure. And this is really essential for mediating the transmission of these complex forces. And so clinically, why do we care about these injuries? They make up about a third of musculoskeletal consultations in the US each year. And unfortunately, in adult mammals, these injuries rarely heal on their own. They typically form very disorganized and mechanically inferior scar tissue. And then enthesis, as I mentioned, because it has a very multi-tissue structure, it often is the most prone site to injury and rupture. And so as you can see in the images for these healthy and injured human pathological sections, as opposed to that nice elongated collagen structure and the cell elongation, you now have aberrant tissue and cellular organization that leads to impaired function. And so, in the case of more moderate disease, if you don't have a full thickness rupture, we often rely on non invasive strategies like physical therapy or in the use of biologics like anti inflammatory drugs. But these often have very much challenges in terms of their administration. And when you have more severe disease states, such as fully torn ends, the gold standard remains surgical reconstruction, wherein you try to take these suture or these torn ends and suture them back together, because the main goal there is to reestablish a mechanical gradient to restore function. But I want to point out that 94% of these procedures often will fail within 2 years post-operation. And a large part of this is due to the fact that we still don't quite understand the biological mechanisms underlying adult enthesis regeneration. And so, as you could imagine, when we're trying to develop therapeutics, it's very hard to do that when you don't know the underlying biology. And so it was really exciting to learn more about Dr. Nabrit's own research program, as our lab also leverages fish models, specifically zebrafish, to study musculoskeletal tissue regeneration. And so early work by our group had pioneered the genetic tools to actually, one, establish that zebrafish do in fact have tendons, and two, that their tendons and entheses recapitulate very key hallmarks and features of the anatomy that they share of the mammalian counterparts. And so, in addition to this, not only do they have very shared structure-function relationships, zebrafish also have their tendons conserve developmental programs that we know. So, the transgene shown here labeling tendon cells is marked by scleraxis A and cherry. So, anything that's a tendon cell is marked by red. And it turns out that during early development in the larval zebrafish, we also see robust induction of scleraxis, which is our most reliable tendon marker. And we also see that into adulthood, you also have conservation of more mature tendon markers like tenomodulin and collagen type 1. And so, other advantages of the zebrafish that make them very amenable for these studies is that they share nearly 3/4 of their genes with that of mammals. And not only that, they're very genetically tractable and they have very high breeding potentials. And so we combine that with their optical transparency, it makes them super amenable for a lot of imaging applications. And lastly, given that we're interested in regeneration, we know the immune response, as the other speakers have kind of set up prior to this, is really integral to wound healing. Zebrafish also possess a very robust vertebrate immune system. So prior work in our group, another postdoc, Marie, had extensively characterized the regenerative cascade of the maxillary superficial tendon, or MST, which is just a craniofacial tendon in the zebrafish. And so this is very advantageous because it's a very accessible tissue, and we can surgically manipulate this by using a microscalpel blade, and we can essentially just cut the tendon off of the maxillary bone and watch it regenerate. So at baseline, as we would expect, when you look at the multiphoton images, we have that very nice collagen ultrastructure, highly aligned, and it's overlaid with these resonant tenocytes and the rad sclerosis M. cherry label. And after 7 days post-injury, remarkably, humans can't do this. So the tendon just spontaneously reattaches to the bone. And this is really key as you want that structural integrity to really orchestrate effective healing outcomes long term. As you would see, 120 days post-injury, these zebrafish tendons can actually fully regenerate their structure and cellular organization. But of course, to study regeneration, it can be very helpful to have a model of non-regenerative outcomes to delineate what these mechanisms could be. And so Marie had also discovered that zebrafish are deficient in tenase C, which are referred to as tenase C mutants, actually don't regenerate their entheses. And so what I hope you can appreciate is that while at baseline they look very similar morphologically, at 7 days post-injury, we already see structural deficits in the healing of these tenase C mutant entheses, wherein there is this gap marked by these two yellow arrows where the tendon has not fully reinserted back into the bone. And while you can't really appreciate it as much here, there is persistent long-term disorganization of the tissue and the cells. And so looking to what tenase C could be playing a role in, tenase C is a glycoprotein in the tendon ECM, and it's known to be dysregulated in a lot of human tendinopathies. But surprisingly, despite this association, we know a lot about what tenase C does in other mechanically dynamic tissues like the heart in terms of fibrosis and pro-inflammatory signaling. But to our knowledge, there are no published studies looking at the role of tenase C in tendon healing. And so to really get a starting point for what these biological mechanisms at play could be underlying this phenotype, luckily at the time I joined the lab, Marie and another postdoc in the group, Steph Tsai, had performed a bulk RNA sequencing experiment of wild type and tenase C mutant samples collected uninjured and 7 days post-injury. And in this case, they had to pull the whole tissue interface of the muscle, tendon, and bone. They're very small tissue samples, so you don't get a lot of RNA. And we subsequently mapped this data to an existing in silico zebrafish matrosome, which gives you the orthologous relations between ECM genes in the zebrafish compared to that of mammals. And so shown here are comparative ingenuity pathway analyses of the biological annotations where in blue boxes in the heat map indicate pathways enriched in the wild-type conditions and orange are enriched in the tenase C mutant conditions. And so the left shows you the uninjured samples and the right shows that 7 days post-injury. And what I hope you can just take away visually is that across the board, all of the processes associated with leukocyte activity or inflammation seem to be enriched in the wild-type condition regardless of injury state. And not only that, when we look at uninjured samples looking at matrosome-specific genes, we see that there is this elevation of proteoglycan gene activity in the tenase C samples. And when you look at in the asterisks secreted inflammatory factors, these all also are enriched in the wild-type condition. So taken together, these shared transcriptomic signatures really implicate a perturbed ECM environment as well as a dampened innate immune response in these tenase C samples. And so for the sake of time today, I won't talk about the ECM, even though that's my child and what I've been working on throughout my Ph.D. and postdoc. I will focus more on the role of the innate immune response. So this led us to the objective of my main project, which were what are the contributions of these early immune subpopulations and their associated inflammatory mediators that may be contributing to adult enthesis regeneration. And so we hypothesized that tenase C deficiency leads to this attenuation of pro-inflammatory activity that then abrogates structural recovery that is necessary to initiate regeneration in the zebrafish. And so just to give you a quick primer, we know that inflammation is really critical in a lot of different organ systems for regenerative outcomes. And in tendon, we know that excessive inflammation leads to fibrosis and aberrant tissue remodeling. On the other side of that, when we have too little inflammation, we actually still get improper tissue repair. And so we actually know that looking at the clinical interventions, when patients take NSAIDs or anti-inflammatories for the pain, these NSAIDs actually have been shown in the clinical data to worsen healing outcomes for patients, and we largely don't know why mechanistically. And so there exists this sweet spot where we want not too little and not too much inflammation, but that very much is unknown, and it's complicated by the fact that most studies of the immune system and tendon have been largely in chronic injury conditions or in developing neonatal contexts or in animal models where they don't regenerate. And so to really address this knowledge gap, we sought to characterize the temporal dynamics of the innate immune response throughout the first 3 days of injury by generating double transgenic zebrafish that label tenocytes and sclerosis M. cherry, or MPEG-1 and MPX-EGFP marking macrophages and neutrophils respectively. And so when we injure these zebrafish, we can collect the whole heads and perform whole mount multiphoton imaging. I'm showing you data for the wild-type neutrophils and the tenase C mutant macrophages on the bottom. And so, as we would expect, there's not much immune cell presence without injury in the wild type zebrafish. But as early as half a day post injury, you begin to see infiltration of these neutrophils in green into the injury site marked by the white dotted lines. And this peaks at 1 day post injury and then gradually declines already by 3 days. Visually, what I hope you can appreciate in the bottom panels is that in the tenascent C conditions or mutant conditions, supporting our hypothesis, we have virtually absence of any macrophage infiltration into the injury site following acute injury, really suggesting that there may be a dampened immune response at play. And so, kind of going back to what are maybe key pathways regulating this response, prostaglandin E2 or PGE2 signaling was one that was identified through our RNA-seq analysis as being downregulated in the tenascent C samples. And coincidentally, this actually is the primary pathway through which NSAIDs actually exert their anti-inflammatory properties. And so zebrafish are a really great model because to study a lot of drug candidates, you just add drugs to their swimming water. And so in this case, we can target mediators more upstream, like COX enzymes that regulate the production of PGE2, or more downstream by antagonizing their prostaglandin receptors, EPs 1 through 3. And so doing a study design in which we just treat the fish with these chemicals, injure them, and collect them for imaging at 7 days post-injury, indicative of that structural recovery. I'm showing you DMS control samples on the left where we expect to see regeneration. And in our drug treating conditions, we actually observed that there are significantly increased gap sizes, as indicated by these increased arrow margins in the receptor antagonist group AH6809. And related to this, we also see that there are diminished SHD intensity, which is usually taken to be reflective of loss of structural integrity, loss of structural integrity. And while the data with the NSAIDs was very much variable, this is not too surprising because it's a very broad-acting drug. And so we did follow-up studies in which we performed more selective COX-2 inhibition, targeting just one of these two enzymes. And we found that this really had a pronounced effect on impairing regeneration in the zebrafish. And I just want to mention that we've also done longer studies wherein we do out to 14 days post-injury after removing the drug, and these phenotypes still persist. So this leads us to a few final questions, one being what does inhibition of PGE2 signaling actually do to the early inflammatory cascade? And so I'm showing you here the same drug candidates where we looked at wild-type neutrophil infiltration. And the main takeaway is that with the receptor antagonist group and the more selective COX-2 inhibition, we have very dramatic blunting of neutrophils throughout the entire healing cascade as compared to their DMSO controls. And the data is very similar for the macrophages. I'm not showing it here for the sake of time, but we see this very robust blunting of any innate immune cells during the early inflammatory phase of healing. And so the last question we wanted to ask was, what were the functional contributions of these immune cells as they recruited to the enthesis and what they were mediating in terms of structural recovery? And so the last experiment we performed that I'm showing today is we can actually use existing genetic lines in which you administer metronidazole or MTZ in the swimming water, and this can actually ablate all of the cells under a specific promoter. So in this case, we did MPX for neutrophils. And so just by swimming the fish in the water with this drug, we can ablate up to 90% of all the neutrophils in the circulation, perform our injury model, and once again, we see that there is abrogation of that structural recovery in the drug-treated conditions. And when we quantify this, while preliminary, we do see that there's in fact increased gap sizes with this drug treatment compared to wild-type controls. And so this tells us that these innate immune cells are required and indispensable for regeneration. So to kind of summarize some of the highlights of this work so far, we're starting to really uncover really key signaling pathways that are seeming to play very important roles in facilitating regeneration of the adult enthesis. And a lot of our ongoing work is doing the opposite experiment where we're trying to rescue this phenotype in the tenascent C mutants by administering recombinant PGE2 mediators. And we're also employing genetic approaches wherein we can use TET-on systems to misexpress immune and ECM genes in a myeloid cell and musculoskeletal cell restricted lineages. So we can see what this role of immune ECM crosstalk is in mediating regeneration. And lastly, just thinking about, I'm an engineer by training, so we think about the clinic and translation. This work really helps us identify also optimal therapeutic administration windows. So we don't have to reinvent the wheel necessarily for therapeutic development, but we can take existing interventions and better guide patients and clinicians in when to actually give them these anti-inflammatories to perhaps expedite the body's natural reparative process. And so with that, I'd like to thank my lab and funding sources. I'm happy to take any questions.
Thank you so much. That was a great talk. We have time for one question in the interest of time. And yeah.
Very beautiful work. I was wondering, do you know if the macrophages present the receptor for tennessine and if that somehow changes their cytoskeleton and polarity and polarizes them towards more pro-inflammatory or anti-inflammatory?
That's a great question. I think that's something that we're beginning to now tease out now that we have evidence that the recruited immune cells are doing clearly something in terms of the ECM response. And so my lab mate Steph, who helped with the bulk RNA sequencing, has a single cell atlas of the enthesis without injury and throughout healing. And so it's actually something we're doing now is looking at there are resident immune cell clusters as well as throughout injury. So we're trying to see which of these subpopulations of immune cells actually are more maybe ECM engaging or interactive. Trying to get at that question you just asked.